speaker is Barbara Merrill. She's working on a tool for visualization of, I guess, biologic networks. Title is Building Networks to Facilitate Metacognition and in Complex Analysis. Thank you.
I'm going to talk a little bit about the meeting of network science and visual analytics here. I am the director of usability evaluation and education and training at the National Center of Integrative Bioinformatics, which is housed at the University of Michigan. And there we develop and research and disseminate interactive visualizations, interactive networks, a lot of them, to help scientists and biomedical researchers analyze huge amounts of data and really find discoveries that have been helping to move medicine and clinical work ahead. In visualizations, interactivity is a huge area, and that's where I'm saying the intersection between network science and visual analytics is, and actually using these networks to explore knowledge for new knowledge. And I'll only look at one aspect of this interactivity, which is interactivity for people's metacognition during their complex exploratory analysis. We've seen already today so much about how visualization and seeing visualization can present, and we can receive, and we can discuss these patterns and relationships that otherwise we couldn't see. But visualizations are also incredibly powerful for the people who are actually doing the analysis. So it's getting it out of the people's hands, like many of you who are making the visualizations, and saying a whole domain should be able to have this kind of analysis to do. And in this talk, I'm going to argue that if network visualizations are going to help people succeed in these kind of discovery flows of analysis, that networks need to bundle visualization techniques for interactivity in combinations that match analysts' actual cognitive tasks and practices. Metacognition as part of this exploratory analysis is seriously understudied in visual analytics today. So in this talk, I aim to expand our understanding of the role of metacognition and its importance in exploratory analysis, and to recognize the supports that networks need to provide in order to help people with their metacognition. First, I'm going to introduce my area of focus, and I'll spend a bit of time on this because I think it's probably unfamiliar to many of you. The metacognitive support that my area of focus needs, though, however, is very relevant across the humanities and social sciences and sciences. I'll define metacognition, and I'll walk through a case exemplifying it in practice, and then based on insights from the case, I'll suggest ways in which network designs and development may better support metacognitive demands. First, my focus on complex analysis for discovery purposes. I'm showing here a pretty well-established model of exploratory analysis in visual analysis. It's the process of making meaning to formulate hypotheses. People start in the blues and greens by accessing and searching for relevant information. Then they recursively interpret evidence, they draw inferences, analogies, and consistently validate all through that. That's the yellow and oranges. They steadily and progressively construct and validate mental models of causal and conditional relationships that may explain some kind of event that is fairly much of a black box. And they look at sets of relationships, and that's in the red. And ultimately, they generate plausible and credible hypotheses. In the research literature on user experience and usability, more is known and understood about the network support that is needed for the reasoning and actions that go on for the greens and blues, the fairly straightforward accessing of information and processing of it, than for the yellows and oranges and reds, which is the more complex, open-ended, ill-structured kind of analysis. But it's in these yellows and reds that metacognition plays a larger role in a domain expert's way of thinking and knowing for discovery purposes, and it's these areas that I'm going to focus on today. Now for some background on the domain and network focus that I'm taking. I examined biomedical specialists and their cognitive tasks in metacognition for one class of problems in biomedical research that relies on network analysis. In this problem, biomedical researchers aim to discover molecular mechanisms of disease processes, often for the purpose of ultimately knowing how to effectively treat causes of complex disease like diabetes or Alzheimer's, and not just the symptoms of them, like we do today. As an initial and non-network phase of this research, scientists typically conduct experiments to find genes that have significant differences between control, healthy groups, and experimental or disease groups. Typically, the result of genes is huge, tens of thousands of them. Now, clearly, not all of these genes can be influential in disease processes or susceptibility to disease. Therefore, the scientists have to reduce the possibilities by finding the functional sets of relationships and interactions that involve some of these genes that suggest plausible and credible influences on the disease. It's highly multiple dimensional and multi-relational. Networks come in at this point. Scientists read the genes or some portion of the genes from their experiments into a network program, like the one that's shown here, which is Cytoscape. The program relates these genes to an integrated, publicly available database of protein interactions, and these interactions are established from other people's research, curated tagging of the literature, and text mining through natural language processing. The program then displays a network of all proteins that interact with the proteins produced by the genes that have been read into the program. The display also shows on the bottom, you can see, a dynamically linked data table that gives the various functional traits characterizing each protein. At this point, researchers begin to explore the network to find relationships of interest. It's a complex, cumulative inquiry that may exceed over 1 or 2 hours at a session. During this time, they use color coding, spatial layouts, selection, filtering, and network expansion to group interactions with similar and complementary relationships and traits that hold some meaning for the disease that they're studying. Their domain expertise is what guides them. And so in this process of exploration, it's the application of their own inner knowledge to the knowledge that they see externally represented. Scientists construct, have open, and compare several networks and subnetworks at once, and you can see that in the green part that's highlighted there. Throughout, they have to keep track of their work and knowledge. They have to maintain a smooth flow of thought amid huge amounts of data. They have to anticipate next steps, and when necessary, recall prior steps and ideas. The goal of a network application, then, is to map support for this kind of complex flow of analysts' work to what their actual reasoning is for this work. So, at our study, we've characterized this process of inquiry cognitively. I've conducted extensive field research and interviews with over 20 users in the field, some of it longitudinal. One workflow in this whole discovery-driven visual analytic activity is finding relevant and potentially meaningful relationships for further study and doing some initial transition into causal and conditional explanations. I'll be talking about just this workflow. My findings show that scientists commonly pursue 4 analytical objectives in this workflow, and for each objective, they tightly combine the same multiple modes of reasoning, which I have listed here. For each objective, they classify in various ways, as shown here. They make comparisons, they apply their metacognition, and they validate throughout. Now, if networks don't adequately support this integrated cognition, including metacognition, then scientists have not been able to complete their work. They have not been able to complete it nor succeed in it, and this has been reinforced by other usability tests in this area that show that scientists do not get the insights that they need for discovery. Often they abandon a program, or they just kind of diminish their goals and say, well, I'm just going to use it to search for information then. So this is a problem. And as you can see, metacognition is a core component of this integrated reasoning. So what is metacognition? You can read the formal definition here. Essentially, metacognition is what we use when we allocate our attention and our cognitive functions as we build, understand new knowledge in a very dynamic and multidimensional workspace. It is vital for fluency and coherence in progressive inquiry, and fluency and coherence is one of the main things that makes an application useful or not to people. Basically what it is it's people's ways of monitoring, managing, evaluating, and regulating their own work as they're going. They monitor understanding of relationships, they maintain an awareness of the context that gives things meaning, they know what to attend to and when, they stay oriented, they know what moves and strategies to use and when, and they also evaluate their own judgments as they go. So let's take a look at how metacognition is part of an inquiry and how it functions. I want to talk about a case to exemplify its role. In this case, a researcher is studying kidney disease. Now, currently, there are many types of renal disease, but there's very little understanding of what makes each type different from one another. And so the outcome of that is that most patients receive the same medication, and that medication is very strong and has very, very harsh side effects. So medical researchers would like to be able to tailor treatments much closer to what your genetic signature is for the disease that you have. And so in this case, the researcher has biopsy samples of 250 disease patients and a control healthy group of donors, and finds 12,000 genes that are significantly different between the two groups. And his goal is to uncover relationships involving these different genes that may credibly suggest some subdiseases, and then make a transition to some causal explanations. He feeds those genes into a program, and now rather than my nice, neat presentation graphic that I have to show you, this is really what he sees, and this is really what most researchers doing this kind of work sees. Okay, so where do you even begin to look into this? And what the objectives are, where I talked about there were 4 objectives. The first objective is usually to analyze the small network that you see, one that seems manageable, one that might give you an idea of relationships that you can then use a process of elimination later with your larger network when you get to it. So he looks at just the small network. And let's look at some of the cognitive tasks that he has just for this small network. What you see in the flow chart are the ways in which these researchers chunk their tasks to move through just this objective of analyzing the small network. And what you see in the text, and I want to just call your attention to the verbs, are things like classify by similarities. That's 1 similarity, 2, 3 similarities, assess and validate as he goes, find and classify by complementary relationships, compare similarity groupings on one trait, secondary traits, and even third traits, manage and keep track of repeated cycling. Now we're getting into metacognition, making binary distinctions of this is interesting or not, this is metacognition, structuring and adding cues to your workspace so that you know where you are and you know what's important, cumulatively recoding items by size and color and manually pulling things apart because you're grouping them as important things because you know that from your domain expertise, keeping track of your data of interest, and deciding what to save or print. These are all metacognitive functions. In my research, colleagues and I have characterized all of the objectives, which I'm not talking about today, and the task chunks, and the cognitive tasks for this workflow, and we've developed a taxonomy of cognitive tasks that characterize this workflow based on what we've seen from user observations and interviews. So to return, what we found is in terms of the types of cognitive tasks, there's these various cognitive tasks, and you can see in metacognition that metacognition makes up a good amount of them. Then what we've done is we have mapped the cognitive tasks that we have, which are 32 is total, and we've mapped them to the various task chunks that they make up. And what we see is that there are many cognitive tasks for any one chunk of work, and a chunk of work is a ways of users' ways of representing their work to themselves. And many of the cognitive tasks are repeated across task chunks, but the interesting thing is that no task chunk has the same combination of them. So what our user studies show is that a small number of cognitive tasks in combination give rise to a large number of analysis functions, and the number of analysis functions in a network grows much faster than the number of features and functions. Attention is a critical resource for this, because how do you make sense and give order to this kind of chaos? You really pay attention to where those data are that you're looking for and the kind of interactivity that you need for them. And what I'm arguing then is that we need to give users control over attention and avoidance of interference by providing very adaptable visualization techniques. So I want to talk just very briefly now of some implications of this, which is designing these bundles of visualization techniques into the networks that we build so that we help people move through their analysis smoothly. One thing that's very important is the notion of helping people see those multidimensional salient relationships that they want to see. So instead of just networks by themselves, a very common visualization technique is this dynamic workplace, so that you've got multiple windows that are all dynamically linked. Instead of just seeing the readouts of certain attributes, what you see in the bar charts are the attributes themselves. They're color-coded. They appear on the network nodes and edges, and they also show, when you select or deselect one of them, they will populate across the bar chart, so you see those kind of connected relationships that people have. For visualization techniques, people rely on perceptual coding, color coding, size coding, thickness of edges. This needs to be made very easy for people. In Cytoscape, it takes 16 steps for you to visualize on an attribute class. And by that time, a researcher's train of thought is pretty well gone. You get these long lists of attributes that go with each node or with each edge, and people want to go through and they want to see which ones are interesting. Right now, in visualization programs, you can usually sort by columns, but what you get is either alphabetical or numerical order. And what people do to highlight where they are, think of yourselves as you're reading through text, they highlight just the line they're interested in to keep their place. What they really want is to be able to sort by their own selected highlighted rows. And grouping and subnetworking is very important, but we have to be able to move from those set subnetworks back to the whole context. So insights for support here are to give these kinds of interactivity in bundles, to anticipate that people are going to apply their expertise and make it easy for them to arrange as they need to and mark their workspaces, and that it needs to be part of network designing and building from the start. I'm going to skip over these and just say that this is very applicable across domains. It helps assure usefulness. It fosters discovery well beyond just reading data off of the presentation and can be enhanced through user studies in your own domain. Before I turn to, I don't even know if there's time for questions, but I had forgotten to put an acknowledgement slide in here, and I'd like to acknowledge the grants that supported this work, as well as two of my co-authors on a manuscript that's being reviewed right now, Felix Eichender and Ben Keller. So thank you.